Vesicles are small, spherical structures. They are composed of a lipid bilayer membrane. Vesicles bud off from cellular membranes. They encapsulate molecules within their interior. Vesicles enclose and protect materials during transport. Vesicles utilize motor proteins to navigate the cytoplasm. Motor proteins facilitate movement along cytoskeleton filaments. This transport system ensures precise delivery of materials. Effective delivery promotes intracellular communication and molecule distribution. Vesicles transport molecules to various cellular destinations. These vesicles navigate to specific target membranes within the cell. Upon reaching their destination, vesicles fuse with the target membrane. Fusion involves the merging of the vesicle membrane with the target membrane. This fusion process releases the vesicle's contents into the target compartment, such as the Golgi lumen. The released materials then undergo further modification and sorting within the target organelle. Golgi apparatus. The Golgi apparatus is a key organelle in eukaryotic cells, responsible for processing and packaging proteins and lipids. It consists of flattened, membrane-bound sacs known as cisternae. These cisternae are organized in a stacked arrangement, resembling a pile of pancakes. The Golgi exhibits structural polarity with distinct cis and trans faces. The cis face is oriented towards the endoplasmic reticulum, where proteins and lipids are synthesized. The trans face is directed towards the cell membrane, facilitating the secretion of processed molecules. Proteins and lipids, synthesized in the endoplasmic reticulum, are transported to the Golgi apparatus. Transport vesicles facilitate the movement of these molecules between the endoplasmic reticulum and the Golgi. As molecules move through the Golgi, they undergo various modifications. Glycosylation, the addition of sugar molecules, is a key modification process. Phosphorylation, the addition of phosphate groups, is another important modification. These modifications are essential for determining the final destination and function of the molecules within the cell. Modified proteins and lipids are processed within the Golgi apparatus. These molecules are then packaged into vesicles at the trans face of the Golgi. Vesicles bud off from the Golgi, encapsulating their specific cargo. These vesicles act as transport containers, delivering their contents to various cellular locations. Destinations include lysosomes for degradation, the plasma membrane for integration, or secretion pathways. This targeted delivery ensures proper cellular function and intercellular communication. Mitochondria. Structure and function. Mitochondria are relatively large organelles found within cells. They are essential for cellular energy production. Mitochondria are distributed throughout the cytoplasm. Their location adapts based on the cell's energy demands. Mitochondria are characterized by a double membrane structure. The outer membrane controls the movement of substances into and out of the mitochondrion. The inner membrane is highly convoluted, forming structures called cristae. Cristae significantly increase the surface area within the mitochondrion. This increased surface area enhances the efficiency of chemical reactions. The double membrane system is essential for mitochondrial function and energy production. Mitochondria are essential organelles responsible for cellular energy production. The inner mitochondrial membrane is the primary location for respiration. ATP, the cell's main energy currency, is synthesized within mitochondria. This energy-generating function earns mitochondria the title of the cell's powerhouse. Lysosome function and structure. Lysosomes are small, membrane-bound organelles found within cells. They are characterized by their spherical shape. Lysosomes contain a diverse array of potent digestive enzymes. These enzymes are essential for the breakdown of complex molecules. This breakdown process is crucial for cellular function and waste removal. Cells can engulf bacteria or other foreign particles through a process called endocytosis. Following engulfment, lysosomes within the cell fuse with the vesicle containing the bacteria. Lysosomes contain digestive enzymes that are released into the vesicle. These enzymes break down the bacteria into simpler, harmless components. Lysosomes are essential for degrading damaged or non-functional organelles. The process involves lysosomes engulfing the targeted organelle. Enzymes within the lysosome break down the organelle into basic components. These components are then recycled, contributing to cellular resource management. Vacuole structure and functions. Plant cells typically contain a large vacuole, often occupying more than half the cell's volume. This large vacuole is essential for maintaining cell turgor pressure. The vacuole also functions as a storage site for nutrients within the cell. Additionally, it stores waste products, contributing to the cell's overall structure and function. Vacuoles in animal cells and unicellular organisms are smaller compared to plant cells. These vacuoles perform specialized functions tailored to the cell's needs. Food vacuoles digest engulfed food particles, a key function. Digestion breaks down food into usable nutrients for the cell's sustenance. Unicellular protists in freshwater environments face a constant influx of water due to osmosis. Contractile vacuoles actively pump excess water out of the cell to counteract this osmotic pressure. This pumping action prevents the cell from bursting due to excessive water accumulation. The contractile vacuole is essential for maintaining the correct internal concentration of ions and molecules within the protist. Chloroplast structure and function. Chloroplasts are organelles found in green plant cells. They are essential for photosynthesis. Each chloroplast is enclosed by a double membrane. This double-layered structure creates a protected internal environment. The protected environment is crucial for photosynthetic processes. The double membrane helps maintain optimal conditions for photosynthesis. Chloroplasts contain chlorophyll, a green pigment essential for photosynthesis. Chlorophyll molecules are specifically designed to capture light energy. The primary source of this light energy is sunlight. The capture of light energy by chlorophyll initiates the process of photosynthesis. This process converts light energy into chemical energy for the plant. Chloroplasts facilitate carbohydrate synthesis using captured light energy. Chlorophyll captures light energy to drive the synthesis process. Carbon dioxide and water are converted into glucose during synthesis. The resulting glucose provides energy for the plant's metabolic activities. Centrioles and microtubules in cell division. The cytoplasm is the fluid-filled space within a cell. It contains various structures essential for cellular function. Microtubules, small cylindrical fibers, are dispersed throughout the cytoplasm. These microtubules provide structural support and facilitate intracellular transport. Microtubules play a crucial role in chromosome movement during cell division. They act as guides, directing the movement of chromosomes within the cell. Microtubules actively move chromosomes to ensure proper segregation. This segregation is essential for creating daughter cells with the correct genetic material. Therefore, microtubules are vital for accurate chromosome distribution during cell division. Centrioles are key structural components found within animal cells. These structures function as anchoring points for microtubules. Microtubules attach to centrioles, particularly during cell division. This attachment stabilizes and organizes the microtubule network. Organized microtubules are essential for effective chromosome separation during cell division. Cilia and flagella. Structure and function. Cilia are short, hair-like structures extending from the surface of cells. These organelles are present in large numbers on a single cell. Cilia facilitate movement for unicellular organisms in their environment. The coordinated beating of cilia propels the cell through fluid. Bronchial epithelium contains cilia, which are small, hair-like structures. These cilia generate a current in the fluid adjacent to the epithelium. This current sweeps mucus and trapped particles away from the lungs. This action aids in respiratory function by maintaining clear airways. Flagella are elongated, thread-like structures extending from a cell's surface. They are generally fewer in number compared to cilia on a cell. A flagellum originates from the plasma membrane of the cell. The primary function of a flagellum is to facilitate cellular movement. Movement is achieved through rotation or undulation of the flagellum, enabling locomotion.